Thank you for attending this lecture today. Good to see you all virtually. So my name is Nate Hipsch. In my lab, we use synthetic extracellular matrix materials both to create better, more predictive in vitro models of disease, and also to instruct cells to differentiate, secrete beneficial cytokines, and otherwise do things that are useful for regenerative medicine. So today we're going to start by talking about, well, why even bother with synthetic extracellular matrices? This is a mechanobiology lecture series, and why don't we get straight to mechanics? I think hopefully you'll appreciate why I think topics like protein fouling are very important for understanding mechanobiology. So first we're going to get into the basics of protein surface interactions. We're going to talk about controlling cell adhesion with surfaces engineered to resist protein fouling. And then finally, we're going to get into mechanobiology, specifically how matrix mechanics affect cell biology. So the first thing we're going to talk about is actually how a surface inside biological fluid will be will interact with protein, and then that protein will mediate an interaction with cells. Now, why do we know about this? Why is this important? Well, from an evolutionary point of view, surfaces, whether they're hip implants or splinters, are a really incredibly good vehicle for bacteria to get into our body and colonize our body. So over millions of years, our body has evolved very robust defenses against foreign surfaces being in the body. From a biomedical and biotechnology standpoint, surface fouling, that is the interaction of surfaces with proteins, leads to leads to rejection of biomedical implants, whether they be pacemakers or hip implants, drug-eluting implants for treating cancer. From a technology standpoint, this causes biofouling on ships, slowing them down in the ocean. This is actually a pretty big, significant problem for the Navy. Now, why does this occur? Well, from a physics standpoint, a material that's put into biologic fluid, or really any fluid, has a high surface free energy. So that surface free energy will be minimized, that excessive surface free energy will be minimized when proteins or dust, or really anything in that fluid, but in this case, proteins, adsorb to the surface from the solution. Now, these interactions of the protein with the surface are going to be due to van der Waal interactions, electrostatic interactions. And when these proteins interact with the surface, especially if that surface is hydrophobic, the proteins will actually change their conformation. If a protein typically proteins have a hydrophobic core. Well, if the surface of the material is hydrophobic, that core will actually be exposed to the surface, denaturing the protein, just because that's a lower energy state for that protein to be in. When these proteins adsorb to the surface, but especially if they change their conformation, they will expose epitopes. These are short strings of amino acids, peptides, that confer specific interactions with cellular receptors. Here I'm showing you the RGD tripeptide discovered by Russellati. This peptide can mediate then interactions with cells. And in the context of a biomedical implant in our bodies, these initial cells will likely signal to more cells and eventually cause walling off of this implant. And again, this is because over millions of years, typically when a foreign material was in the body, this was going to be something like a splinter that would be a good vehicle for bacteria. So it's in the best interest of our body to wall this off and protect the rest of the system from it. Because of the importance of protein fouling and protein surface interactions in cell biology and biomedical materials, there's a large number of techniques available for characterizing this. And I would say very broadly, I would group these into two categories. There are techniques that are fluorescence-based techniques like TURF. There are techniques where we radio-label our proteins. I-125s are probably the most common way of doing this. And then there are antibody and cell-based assays as a second category. Now, in the first category, where we label the protein molecules of fluorescence or radioisotopes, what I'd like you to appreciate is that the assay itself is relatively agnostic to whether the proteins are in a conformation that's bioactive, that is, they're exposing this triangular epitope. This represents a string of amino acids, again, a peptide that interacts with receptors. So these techniques can't really distinguish between exposure of active epitopes and protein that's there but not active. In contrast, antibody and cell-based assays are relatively insensitive to proteins that are in a conformation where the active peptide is not available. So you can see here, this protein molecule happens to be presenting the active peptide on this surface where it's not available to a cell receptor. This protein is also denatured, and in this case, denaturing of the protein is causing this peptide epitope to not be bioavailable. So again, total amount of protein, bioactive protein molecules.
This mild detergent is going to permeabilize the cell membrane and get rid of things that aren't interacting with the cytoskeleton. However, the cytoskeleton will be left intact. So this allows us to differentially extract things interacting with adhesions and things interacting with the cytoskeleton. Now we can take this one step further. If we use very high amounts of salt and high amounts of detergents, we're going to wash away even the cells, even the cytoskeleton, but not the extracellular matrix. The extracellular matrix is very poorly soluble. So just to give you an idea, this procedure here, this is a 5-minute wash in PBS. This is a 5 to 10-minute exposure to cold lysis buffer. To actually extract this dense extracellular matrix involves boiling in very, very high concentrations of SDS, like not 1%, but 10%. So historically, biologists and bioengineers have used these, taken advantage of this differential solubility to selectively look at different parts of the cell and different parts of the extracellular matrix. Now we're going to delve into some use of these techniques to actually assess protein adsorption to surfaces and how cells behave on them. And we're going to start with a biomaterial that probably most of you have used. If you've done cell culture, you've used an engineered biomaterial, which is tissue culture polystyrene. Tissue culture polystyrene is generated by taking regular old polystyrene and exposing it to oxygen plasma. This creates functional groups on the surface that favor protein adsorption. So you can see here on the left, if we quantify using radioactive protein, the amount of adsorbed vitronectin as a function of the input vitronectin on regular versus tissue culture modified polystyrene, you can see that this tissue culture treatment dramatically enhances vitronectin adsorption. Fibronectin adsorption, there's a trend towards it being enhanced, but it's not significant. So already you're seeing that the surface, irregardless of the ECM protein you add, the surface itself under the ECM can have effect on the ECM that you actually get, regardless of what you put in. Now, if we look then at cell behavior, so we're going to use the I-125 radioactively labeled protein to give us an idea of the total amount of protein on the surface, and then normalizing for differential adsorption, we're going to compare cell adhesion on vitronectin that's either adsorbed to regular polystyrene or tissue culture polystyrene. And with a couple of different cell types, we can see differences in these curves. Now, just by show of hands, maybe you can give me, let's see, maybe you can give me a thumbs up for tissue culture polystyrene and a clap for regular polystyrene. Which of these surfaces is better for promoting cell adhesion through fibronectin or vitronectin? Is it the black regular polystyrene, or is it the open tissue culture polystyrene? So tissue culture polystyrene, give me a thumbs up. Regular polystyrene, give me a clap. Okay, I've got 4 thumbs up, 5 thumbs up, 6. Okay, good. This is very important, everyone. You all need to get used to voting. All right. So yes. So even with a constant density of protein on the surface, the tissue culture plastic is better at presenting the protein to cells than regular polystyrene. This is another classic experiment in cell biology. This is work from the 1980s from Fred Grinnell. And in this case, Fred's group was trying to study kidney cells outside the body. And in fact, a lot of early insights into how biomaterials interact with cells comes from basically this era before mouse genetics really took off and we were forced to culture cells. And a lot of technical and scientific work was done to figure out, well, how the heck can we keep these cells happy outside the body? And Fred and others had found that coating fibronectin on polystyrene did not elicit cell adhesion and spreading. But if they actually primed the polystyrene with a layer of fibrin, so basically a blood clot, they were getting robust spreading of the cells. So in this paper, they're using CIG. CIG was an acronym that means fibronectin. So I'd like to have maybe one of you can, maybe 1 or 2 of you can just volunteer and answer about what are 2 possibilities as to why the CIG, when we go ahead and cross-link the CIG on top of the fibronectin, is giving us far more cell adhesion with less CIG added than if we just take the CIG fibronectin and put it on the plastic. In other words, why do you have to put in fewer of these red fibronectin molecules with the priming layer of fibrin than without it? So anyone want to volunteer? Maybe it's a small group, so you can just unmute yourself and speak up.
Is the fibrin more likely to bind to the fibronectin is more likely to bind to the fibrin layer than the polystyrene, so you need fewer?
So I'm having trouble hearing you. I think Christian's talking. Could you just type it in the chat? Thanks. Yeah. Okay. Chat. So, yeah, it relates to charge. There's kind of there's two possibilities. Anyone else want to volunteer? So Christine is saying. So Christine is saying the cells recognize the proteins and are more attracted to the coated surface. That's a good thought. And then Christian Tessman is saying, well, maybe the fibrin is more likely, the fibronectin, the CIG, is more likely to bind the polystyrene than it is to bind, or sorry, maybe the fibronectin is more likely to get stuck down on the fibrin than it is on the polystyrene. In other words, if I go back here, maybe the fibrin clot is more like tissue culture polystyrene, where it's just better at sucking up fibronectin from the solution and depositing it on the surface. So now we'll go forward to some more data from this paper. So what this data is showing you is actually if we measure the amount of CIG, this is, again, CIG they're using as an acronym for fibronectin. If we look at the amount of CIG bound to plastic versus bound to fibrinogen, what we find is that the fibrinogen is actually worse at taking up the CIG and binding it than the plastic. Look at how much they're having to add to get a few units here versus how much they're having to add here to get an equivalent number of units. So the fibrinogen is, the blood clot is much worse than plastic at absorbing fibronectin. So with that in mind, what's another possible explanation of why this surface that's sucking up less of the fibronectin is doing a better job at promoting cell spreading and adhesion than this other surface? It looks like Sabrina is trying to unmute. No. Okay, so just to, I guess to move forward, the cells don't necessarily interact with all of these fibronectin molecules. The cells only interact with fibronectin molecules that are presented in the right conformation. So the receptors of the cells don't just recognize that the protein is there. They recognize specific small groups of amino acids called peptide motifs. And if the fibronectin is absorbed in a conformation where this particular group of amino acids, this peptide motif, is not available to the receptor, then from the cell's point of view, the fibronectin might as well not be there at all. Okay, so this antibody-based assay is much more representative of what the cells would see than just looking at the overall amount of protein on the surface. So if I go forward here, now we're going to go over another kind of classic paper in ECM cell biology. This is a paper that was written by one of the more famous researchers in cell adhesion and integrin-mediated biology. This is from Andres Garcia, who's at Georgia Tech. And Andres was interested in the late 1990s in the observation that if you wanted to take myoblasts and study their conversion into tubes, these muscle tubes that stain positive for these green muscle proteins with this antibody assay, it was better to culture them on fibronectin-coated collagen than on either regular polystyrene or tissue culture polystyrene. So you can see the cells can grow just fine on all three of these surfaces, but with the same environmental cues, the induction of the myoblasts into muscle-like tubes is much, much better when, again, you have that priming coat of collagen on the surface. So this is some data from Andres Garcia's paper. And again, we can measure using radioactively labeled proteins the amount of protein on the surface. And here we can see the collagen gel is worse than the polystyrene in terms of its ability to present lots of fibronectin. Both polystyrene surfaces are pretty good, at least in this assay, at absorbing fibronectin. However, when different monoclonal antibodies are used to probe the surface for, again, remember, this is looking at the total amount of protein. These are looking at the presence of specific peptides on the proteins. These different antibody-based assays are giving us a very different response curve where at a constant density of fibronectin on the surface, for this antibody, the bacteriological, this is the unmodified polystyrene, is much worse than tissue culture polystyrene and much worse than collagen at actually presenting this peptide. And again, in the interest of time, I won't pull you on this one, but this assay is measuring bioactive peptide motifs. The antibody can't bind to the peptide if it's on a part of the protein that's buried in the interior core. It can't bind to the peptide if its peptide is facing the surface rather than facing away from the surface. This assay is agnostic to the bioactivity of the protein. So this assay would be kind of like you open your refrigerator and you're thirsty and you want a certain kind of soft drink, and you're scanning for that kind of soft drink, and you're scanning for a full bottle of it. This is, you're detecting all bottles of any liquid in your refrigerator. Anybody want to comment or guess on why do these different monoclonal antibodies have these different response curves for the surface presented fibronectin? Check the chat. So I guess maybe a yes. So yeah, Christine is saying the antibodies look for specific markers. Yeah, and Gabriel is saying the peptides are on different parts of the protein, so that you're both right. So these antibodies recognize specific peptides. And because these are monoclonal antibodies means that each antibody only recognizes one peptide. So this particular antibody and this particular antibody, they both recognize specific parts of fibronectin, they just recognize different parts. So moving on, Andres's group, again using this differential solubility approach, they took cells, allowed them to adhere to these different surfaces, and then used a chemical crosslinker to force integrin adhesion receptors to be bound permanently to any ECM molecule they're engaged with. This means that they can now use these very harsh solvents to wash away all of the cell except for that integrin that's actually bound to the ECM, and they can then quantify the amount of integrin present. So this is the amount of each of these integrins that are actually bound to a constant level of absorbed fibronectin, when the fibronectin is presented from different surfaces. And it's a little hard to see, but here on these, but when you quantify the density of these bands, so these bands are obtained by doing a Western blot for bound receptors, what you can see is that the amount of this alpha-5 integrin receptor is higher when you have fibronectin presented from a collagen priming layer or a tissue culture plastic than from bacteriological plastic. So what this is telling us is that the antibody assay is a lot better than the radioactively labeled ligand at predicting how cells will behave. Because like antibodies, cell receptors, they don't care about the overall amount of protein, they care about the overall amount of bioactive peptides being presented from the surface. So in this little module, I hope I've convinced you, well, why do we need to bother thinking about synthetic ECM in the first place? Well, for starters, it's very wrong with extracellular matrix to assume that what you put into the reaction or what you put into the culture vessel is actually what cells see. Surface chemistry has a really strong effect on the bioactivity of ECM, and this stems from our need as organisms to fight off infections. So the fact that surfaces interact with different ECM proteins in different ways allows our body to recognize a foreign surface like a splinter and be primed to attack bacteria that were likely carried into the body by that foreign surface. Another very important concept here is that when we're using full ECM components, we might not even have an interaction between the integrin receptor we care about and the ECM protein we put into the system. ECM proteins interact not just with adhesion receptors, but one another. So this, I hope, the three of these reasons together make you at least consider why synthetic ECM may be important looking into mechanobiology. So now we're going to talk a little bit about controlling cell adhesion with fouling-resistant polymers. So again, I mentioned that our body doesn't care if we have a splinter, it doesn't care if we have a cardiovascular stent or a hip implant. Our body recognizes foreign surfaces as being immunogenic and inflammatory. And there have been decades and decades and decades of research to try to circumvent this. Because you can imagine, while this is a very good thing for us to have evolved when we're trying to not get infected and die when we get a small cut, it's a very bad thing when we need a cardio stent or when we need some kind of implant. This causes implant failure. And this is a subject that's been studied for, I would say, close to 100 years now. This is just a cartoon from David Granger showing not just some proteins interacting with the surface, but really a really dynamic response where there's different cells over different times. And this all starts with protein adsorption to the surface. So an obvious question from a physics and engineering standpoint is, well, what if we just prevent the protein from sticking to the surface? And indeed, there have been very extensive efforts over a very long period of time to try to basically modify surfaces to make them less likely to absorb proteins. So this is an example of a biomaterial. This biomaterial happens to be the type of material you would use for a pacing implant, so PTFE. So when this is coated with a material called tetraglime, you can see at low magnification and at high magnification, we have far fewer cells sticking on the surface than if it's unmodified. And you can see this is just a quantitative assay looking at adhesion of platelets. These are the cells that form blood clots. And in all cases, modification with this material tends to inhibit cell adhesion by inhibiting protein adsorption. Now, for those of you that have taken biomaterials classes, you know that long-term, it's very, very, very difficult to prevent any cell adhesion of the surface. This is why right now that if we go below about a 3 to 5 millimeter size of a vascular graft, we cannot use a synthetic material. And we still, for a cabbage procedure, we have to take a vein out of your leg and put it into your heart. It's because 60 plus years looking at this, we can't prevent the adhesion. However, over short time frames, and in an in vitro environment where we don't have an immune system around, we can take advantage of surfaces that minimize protein adsorption to take away these non-specific protein adsorption, non-specific changes in protein conformation, and really dial in adhesion to our at will. And because of their physical chemical characteristics, they're hydrophilic and they also swell well with water. Hydrogels are very good at minimizing non-specific protein adsorption. And on a very basic level, what this allows us to do is we can encode stiffness, we can encode ligand density, we can encode these things independent of one another, but probably just as important, extracellular matrix proteins from the environment are unlikely to stick to this protein. So that just means from an experimental design perspective, what we put in is a little bit closer to what the cells actually see than it is either when we use natural ECM, which can bind other ECM proteins, or if we use surfaces like polystyrene. Another challenge that comes, even if we are using these non-fouling materials, is that extracellular matrix molecules are complex. Extracellular matrix molecules interact with other ECM molecules, they interact with growth factors, and they interact with proteins that cells make, called matrix metalloproteinases that can actually chew up this ECM. This is just a map of fibronectin. Fibronectin has many active peptide domains, and these are kind of highlighted here. So what's one solution to this? One solution to this is to not bother with the full-length ECM protein necessarily, and really just focus on bioactive peptide motifs alone. And in this case, this is a seminal work from Erkki Ruslahti and Michael Pirschbacher. This is showing that you can basically recapitulate the adhesion function of fibronectin and allow cells to spread if you take small peptide fragments from fibronectin. So in this case, they mapped the adhesive site of fibronectin to a tripeptide called RGD. And RGD alone, it's not exactly the same as fibronectin in terms of eliciting cell adhesion, but it's much better than not having any peptide at all. And what I'd like you to appreciate is if we simply take out this RGD motif, then we completely abrogate the adhesive activity of the protein. Or if we change one of these active amino acids, so if the glycine is changed into a valine, so going from RGD to RVD, we completely wipe out cell adhesion. So these are examples now of combining a bioactive peptide with a non-fouling surface to control adhesion. And from this, you can appreciate qualitatively as well as quantitatively that over time, without this adhesion ligand, very few cells can engage this surface. Their ability to engage the surface initially and proliferate on the surface over time depends on the presence and density of this tripeptide. And one thing that's important here is that this is done in the presence of serum. So on the plastic, even though there's no RGD added, proteins from the serum like vitronectin can stick down and be presented. Cells, the ECM proteins that the cells make over time can also change their adhesion and proliferation rate. But with this biomaterial that inhibits protein adsorption, we really have far better control over non-specific adhesion. So we know that the only thing the cells are really sticking to in this system is this RGD tripeptide. And here I'm just showing you examples of using different non-fouling hydrogel materials. Again, these are gel materials that are good at resisting protein sticking. So this is sodium alginate. This is a material we use in my lab. By itself, very few cells adherent with the RGD tripeptide. Almost acts like a normal extracellular matrix. Similarly, PEG diacrylate hydrogels do a very poor job at eliciting cell adhesion and spreading unless they are modified with this same tripeptide. So now we'll talk a little bit about what happens after RGD engagement. And I think you've probably covered this in other lectures, so we'll go very quickly through this. But upon engagement of integrins with the ECM, we get clustering of integrins, so multiple of these receptors, typically in a heterodimer like this with an alpha and a beta subunit. This clustering recruits focal adhesion plaque proteins, and this causes the integrins to be linked to the cytoskeleton. So now we have a mechanically contiguous linkage. And in fact, people have done studies that show that once you have integrin engagement, if you pull on this integrin, you pull on the cytoskeleton as well. Another interesting thing I mentioned that you need clustering of integrins. Well, by having this surface that's good at avoiding protein adsorption, protein sticking, you can look at the effects of peptide density. And what some investigators have done over time, this is seminal work by Masha and Jeff Hubble from the early 90s. You can see that as you dial up the concentration of fibronectin, even though the cells can spread at a threshold density of the CCM protein, you can look on the interior of the cells and see an absence of these F-actin stress fibers. So what this represents is a cell that's sticking down, but a lack of integrin clustering and a lack of robust focal adhesion assembly. And from a translational standpoint, sometimes we actually want this for certain signaling programs within the cells. We don't want stress fibers to form. In multipotent cells, the stress fiber formation tends to give us a more fibroblastic phenotype. This tends to give a less fibroblastic phenotype. And again, this can be done by preventing non-specific protein adsorption and then dialing in the appropriate amount of adhesion molecules to get some spreading, so less of a spindle shape here than here, but to not get stress fiber formation. Even in adhesion independent contexts, like if we have the cells encapsulated in a 3D environment where they're stuck with gel all around them, certain gene expression programs, for example, osteogenesis, so turning on a bone differentiation pathway, are favored by integrin engagement. So these are just examples from two independent groups. This is from Jennifer Alicia, this is from Christy Ansett, showing the amount of bone markers like osteocalcin, CBFA1 RUNX2, this is a mineralization stain, they all increase in proportion to the amount of RGD. So as you're probably going to see in other lectures, ECM integrin engagement not only allows the cells to survive, not only allows them to spread, but allows them to be receptive to different soluble cues that cause differentiation. So just to summarize this section, minimizing that non-specific protein adsorption is very important, if for no other reason than allowing us to at least say, what I put in is pretty close to what the cells see. The ligand, the type of ECM ligand, so the RGD tripeptide versus other peptides and its density, are important for controlling cell responses. And mechanically oriented responses like stress fiber formation depend very significantly on the type and density of ligand. So any questions on these first couple of parts before we move on to the last part of the talk? Okay, so I see there's no questions, so I'll go ahead and move forward.
There's a question in the chat.
I'm sorry, could you type your question in the chat? I can't hear you very well.
So the question is from Chris, and he's asking, what advantage is there to using RGD motif than simply adding collagen to functional?
Sorry, for some reason I really can't hear you well at all. Could you type your question in the chat, please?
Yeah, so there's a question in the chat.
Oh, okay. Yeah, so what is the advantage to using the RGE motif than simply adding collagen to functionalize your material? That's an important question. So let me go back here. So in this paper that we went over, collagen is used not as a direct way to engage integrins. It's used as a priming, almost like a priming coat to prime the adhesion, the adsorption of fibronectin in a bioactive pro-signaling conformation. So I would say one very significant disadvantage of just using full-length ECM protein, particularly collagen, is that the actual receptors that are involved in transducing mechanics or synergistically signaling with growth factors or something else, those might not even be collagen receptors. Those might be receptors for a protein that's sticking to the collagen. And remember, these ECM proteins don't just engage with integrins, they also engage with other ECM proteins. Did that answer your question?
Yeah, that did. Thank you.
Okay. I'll hope I answered your question, and we'll have a little time at the end in case I didn't. Okay, so now moving on. This is a mechanobiology lecture series after all. So mechanical properties of the synthetic ECM affect cells. And again, starting with some historical work, this is seminal work by Don Ingber. This is work by Dave Mooney. What this work by Don Ingber shows is that to have cells respond properly to fibronectin, it doesn't just matter that the fibronectin is available to engage receptors. It matters that cells can exert tension on the fibronectin. So in this experiment, Don used tritiated thymidine incorporation. This is an assay for proliferating cells. And he looked at cell proliferation in response to the availability of fibronectin. Now he presented fibronectin either as a coating on a dish. This allows cells not just to stick to the fibronectin, but actually to pull on it and exert tension and spread. Or he presented fibronectin in a soluble form where it could only bind at the top of cells or on beads. And what you can see here is that the bioactivity of the fibronectin, as we went over before, it depends on the surface it's presented from. It also depends on the cell's ability to exert tension. So to directly address this idea that exerting tension and the ability of the surface of the fibronectin on to resist tension is important to cell biology, another researcher, Yuli Wang, did a very clever experiment. This is taking one of these non-fouling hydrogel materials. This is taking bisacrylamide. And this is changing the stiffness of the bisacrylamide, going from a very soft gel here. And you can see this is a very, very rudimentary way of testing mechanics, basically putting a weight on it and seeing at steady state how far the material stretches. And comparing the cell response on this to the cell response on this, I will say that this kind of alludes to the question Chris just asked. This was done by taking the bisacrylamide and coating it with collagen. So we can't be 100% sure if the cells are actually interacting with integrins, through integrins with collagen, but let's assume they are. Compared to a soft material on a stiff material, the cells are able to spread more. So this is a soft versus a stiff material. But also if we look at signaling, so if we look at phosphorylation, this is a marker of, generally a marker of signal activation of focal adhesion kinase, FAK, and also the presence of paxillin, a focal adhesion complex protein. What we can see is on the soft surface, not only is there less of this focal adhesion contact protein there's also less signaling. So even though the soluble environment is exactly the same, the cell's ability to receive a signal from that collagen depends on the ability of the material presenting the collagen to resist cell traction forces. This is classic work by Adam Engler and Dennis Disher showing that cells' ability to spread depends very dramatically on the stiffness of their environment. This is again using a collagen-coated bisacrylamide gel. And finally, if we look at cell lineage and differentiation, in the CEMB program, I have no doubt you've probably seen this paper, and if you haven't, read this paper. This is a very important paper to the bioengineering field. Both in 2D with cells on top of gels, and with some historical work that I've done in 3D with cells in gels. Cells don't just change their adhesion and spreading, they also change their fate in response to stiffness. So here, Engler and Disher show that going from multipotent cells that can convert to nerve, muscle, bone, fat, in soft environments, we tend to get very different fate of the cells than in stiffer environments, even if all the soluble hues are constant. And from a translational perspective, we can actually take advantage of this. If we transplant cells and materials with the right stiffness, they can actually not just give us a bone-like differentiation, they can give us contributions to bone formation within injury sites. These are within cranial skull defects. So I'll skip this in the interest of time and just get to some current issues, and then hopefully have some time to answer questions. Timing is important here. So I skipped the slide on stress relaxation, but also there's another question of timing. How long do cells need to be in a stiff environment to respond to it? In vivo, there are many situations like injury and fibrosis where tissues stiffen over time. How does the dynamics of the stiffness affect the cells? And then what are the signaling mechanisms? I think one big challenge in creating synthetic extracellular matrices is that we have a massive variable space. So I kind of glossed over, we can control ligand density, we can control ligand type, we can control the nanoscale distribution of the ligands, we can control stiffness, stress relaxation, and then when we get into things like 3D, we can think about pore size. We can think about whether the matrix is plastic or more elastic. So it's a massive variable space that's rather hard to address experimentally. And one of the challenges when you have this big variable space is if you're undersampling it, it's very easy to get tricked into thinking that certain variables are not important. So in other words, if my goal is to show that stiffness really doesn't matter, and the density of adhesion peptides matters, well, I know that if I pick material, if I pick a certain stiffness range that's not very relevant to the cells, then the cells won't respond to stiffness and they'll respond to the ligand density. This raises the question, well, how should we test these different variables, and how can we use that to understand the way cells engage with natural ECM? I think one way is with combinatorial approaches to designing materials that allow us to construct material libraries and test many variables. But I would argue that another important thing is to really use these materials to ask translational questions. To ask, for example, well, is the soft material going to help us either by transplanting cells in it, or is it going to help us expand stem cells outside the body? The reason I think this is important is this forces us to put ourselves in a part of the variable space that's physiologically relevant. Also, you can imagine here, this 12 kilopascal surface seems to be better at expanding these muscle stem cells than this really soft one and this much stiffer one. But you can imagine we could probably do better by adding in different soluble cues. Identifying the things that improve the translational outcome will tell us something about the basic mechanisms cells are using to sense this environment. And I'll just leave you on this slide. I would say another final comment on this is that when we're using synthetic extracellular matrices, we have the opportunity not just to ask fundamental questions about how cells sense their environment and how mechanics are transduced. We also have the opportunity to ask, how could I make a material that's easier to incorporate in a microfluidic device, or a material that's easier to inject? For example, natural extracellular matrices tend to be very viscous. They tend to be very temperature sensitive. With synthetic ECM materials, even if it's not a knob that the cells care about biologically, working time, that is time between putting the cells into a system and cross-linking it, is very important from a functional standpoint. And this can be one thing that guides our design of these materials. So with that, I apologize for the trouble logging in initially, and I'm happy to take any of your questions in the chat.
Thanks, Nate. Those of you who have questions in the chat, just go ahead and ask them.
Yeah, sorry, I can't, if someone's talking, I can't hear you. Could you use the chat? Can cells, yeah, so Chris wants to know, can cells migrate in 3D matrices? Yes, yes, they can, given the following. So if we put cells into a material with very small pores, here you can see these are cells encapsulated in alginate gel. Here, the pore size is a lot smaller than the cells. The cells are on the order of 10 microns. The pore size is on the order of 5 nanometers. So they're not going to be able to migrate through this. However, there are a few ways we can allow cells to migrate through these materials. The first way is to have the material partially degrade. So as the material degrades, that will create voids that are large enough for cells to migrate through. The second way is we can, we can take advantage of the fact that in natural ECM, there are these matrix metalloproteinase degradable sites. So there are peptides that elicit adhesion. There are also peptides that can be degraded by specific enzymes. So if we cross-link our material using these matrix metalloproteinase responsive peptides, that's another way to elicit cell migration. And a third way to elicit cell migration is to make the material not be a purely linear elastic material. If we make the material have some plasticity to it, so that cells mechanically can induce a permanent deformation in the material, then through their traction forces, the cells can basically open up pathways for migration. Okay, so I think I've answered your question. Any outstanding questions before the next talk? Okay.
Yeah, looks like we're done. Thank you so much, Nate. Thanks for a great talk.
So with that, I think I'll go ahead and sign out, but we're really excited for you all to be involved in Center for Engineering Mechanobiology. Thanks to Ron and Patricia and everyone for putting this together, and excited to see you all, whether virtually or non-virtually, at some point in the future. So thank you all.